Hello, everyone. Thank you for joining us today. And this is actually the first webinar in the series related to creation and exploration of ultra-large trillion-sized chemical spaces. So before we start the presentation, this presentation, I would like to remind you what ChemSpace is and what we're doing and why we actually have the interest to this huge chemical spaces. I hope you can see my screen now. ChemSpace role is broader than it's been before, and it's related to the discovery of small molecules or hits through a variety of different approaches with further support into the procurement, sourcing, as well as compound management. And as we all know, chemical spaces is enormous. 10 to the power of 63 molecules is really something that we can't really imagine. But out of that chemical universe, we need not just the molecules, but the samples we want to screen. And those samples can be effectively delivered by ChemSpace out of 7.7 million in-stock items and trillions of make-on-demand compounds. And when I say make-on-demand compounds, I mean not something that can be actually imagined, but indeed synthesized by our respectful chemical partners like Enamine. And obviously, once we have those variety of molecules, either from in-stock sources or from make-on-demand sources, we are able to screen them, to find the hits, or to get analogs for your hit molecules. And those are just few options that we offer. And more about the chemical spaces and the tools you'll be able to hear from today's talk and from our today's presenter, Vsevolod Seva Katritch, who is associate professor at University of Southern California. And today he will be talking about readily available chemical space and everything related to this. So with this, Seva, I would like to pass the word to you.
You know, it's my second presentation at ChemSpace, one we did last year, and it's always a great honor and pleasure to present to ChemSpace and Enamine crowd, because you guys basically created this field of on-demand libraries that help us to expand like our screening to spaces that we were never like even dreaming of. And also I'm really want to emphasize the role, and I'm really amazed at the work and heroic work that ChemSpace and Enamine doing this time, scaling up and working very reliably and expanding this area quite a lot. So and that's why today I will talk a little bit about our work on V-SYNTHES, and also how it is scalable to trillions of compounds and beyond to match the growth of the libraries here. So I will start with like humble reminder. I use the words drug discovery in my first slide. So, and some people chuckle at sort of the notion of drug discovery being done like in academia. Well, we understand the enormity of the drug discovery process itself. More than decade of efforts of like very different sites and like billions of dollars of investment and multi-stage. So where we contribute is like the drug discovery by itself. So hit discoveries, hit to lead, lead optimization, and hopefully some computational work is done in preclinical development that sort of helps this. To make the drug discovery picture like even more intimidating, this is the full map from National Center of Advanced Technologies, NIH, like showing all the steps in this drug discovery. So it's colored according to this pipeline. And again, we are here in hit lead discovery and optimization. What is also interesting at like in this, it's like 5-year-old version of the map, and the only places where computational is even mentioned is these two. So cheminformatics on lead identification, and then some in silico modeling of lead compounds, so like QSR probably. So we will try to like show you today that that's changing, and there's much a bigger role for computational tools in drug discovery, and that's drug discovery can be actually computationally driven. Okay, so my lab came into drug discovery from like studying structure and function of GPCRs, specifically focusing on this family for about a decade, and developing 3D modeling tools for analysis of the structure and connecting it to function. And also in parallel, we found out that we can use the information derived sort of from structure function analysis. We can use in application to ligand design. And some of the first applications were rational design, and we still do a lot of sort of semi-manual design of derivatives from known scaffolds, like this morphinon scaffold, for example. But also what is like more exciting and where our focus shifting now is virtual screening on GPCRs. And basically we, like in the last couple of years, we go way beyond GPCR family and expand it to like all clinically relevant targets. Some of the core computational technologies that we are using is internal coordinate mechanics coming from MolSoft, and so there is an acronym ICM for internal coordinate mechanics. It represents the atoms of the molecules and the molecules of protein and molecules atoms of the ligand. It represents them in internal coordinates, internal chemical coordinates, bond lengths, bond angles, torsion angles. And this makes, unlike Cartesian coordinate, it makes all the movements in this space are comply to sort of chemical geometry, and it's much faster and much easier. You remove a lot of coordinate that too rigid to make difference, basically focusing on torsion coordinates. So this speeds up convergence when you need to find minima. It speeds up like all sampling 1,000-folds. Molecular docking is also based on this concept. So ligand is fully flexible and presented in internal coordinates. It can tumble in the pocket and can rotate. The torsion coordinates can rotate. A receptor is simplified even more, represented by grid potentials. So This gives us several orders of magnitude of speed again, and allows very fast sampling, and also introduces some like additional softness in the receptor, sort of flexibility, implicit flexibility that is helpful, because otherwise the system is too sensitive to very minor changes. Molecular dynamics is a great tool. It's like pretty heavy for GPCRs. You need to put them in membrane and solvent, and it's like hundreds of thousands of atoms. So it is quite heavy and slow. You can't use it for docking, for example, but you can explore some kinetics of molecules. For example, you can see here a lipid molecule from membrane walking into the binding pocket of cysteine-leukotriene receptor. So like opening the gate between helices 4 and 5 and just like walking straight into the pocket without leaving membrane. So you can get like very interesting insights into how ligands bind, how receptor changes conformations, and all these sort of connection between these two things. We also rely quite a bit of our research on bioinformatics, cheminformatics, and more recently we introduced some elements of machine learning. I will talk a little bit about this. So I don't want to dwell on tools specifically, but like core tools, but rather to explain sort of where we are, what is our like main platform. So today I will talk mostly about V-SYNTHES. First, sort of rehashing again for those who heard this talk already. Sorry, it will be pretty similar. So about a third of the talk will be about the V-SYNTHES, our first paper a year and a half ago. And then I will talk about new developments and new applications of V-SYNTHES. And then like a little bit more philosophical sort of part of our vision of computational driven drug discovery. So where we are going, what the next bottlenecks. Okay, so diving into V-SYNTHES, just to introduce the concept itself of virtual screening. If you want to discover new hits and leads for protein target, you usually start with a high throughput screening, which is expensive and slow, and there are like quite a few drawbacks. In primary hits are very often not reproducible, so it's still viable way of entering, but there is also virtual ligand screening, and we're focused on structure-based virtual ligand screening when you have structure, but you don't have much information about previous ligands. So the way to do virtual screening is just to dock every molecule into the binding site. So you see this mess, like with all molecules docked here, and select the best for. And you can do it with tens of millions of compounds, focusing, selecting a small focused library of like from 10 to 30 to 1,000 compounds that can be purchased or synthesized and screened experimentally. So this reduces the size of experimental screening enormously, and also allows to expand into molecules that are not on shelf, right? So it's when it works, it's faster, cheaper, better, and we show that it works with pretty reasonable like hit rates, and our experience over a decade with probably like a dozen of different projects, many of them published, showing like 20 to 40% hit rate. So if you select 100 compounds for screening, for testing, like 20 to 40 of them would be hits. but recently, this whole field sort of got a huge boost due to several sort of factors. One of them is availability of targets. There is structural genomics made huge progress in the last 20 years, and keep making it with cryo-EM boom, and also with AlphaFold models, which is a little bit sort of still exploratory part, but still basically for every target you have a decent structure to start with, and a lot of high resolution structures that are really sort of good for virtual screening. There was no like big revolutions in development of tools like docking and screening, but there was a very sort of robust and continuous evolution with scoring function being improved, sampling methods being improved, like introducing new speed up sort of algorithms. So pocket optimization to validation and optimization to make sure that we dock in the right structural model. So all this sort of happened in the last 15 years, and it brought us closer to sort of more reliable virtual ligand screening. So I already mentioned ICM. So the ligand, flexible ligand docking algorithm there is very robust, very fast. So for drug-like molecules, like less than 500-600 Dalton, it converges in 5-15 seconds, finding the binding poses of ligands. So it's all energy based, and it's sort of generalizable. You don't need to re-optimize the function for each target. This and also ICM won several recent challenges, like blind challenges, benchmarks for docking and screening. It's called D3R. Okay, so and I also mentioned optimization and validation of receptors. So you can optimize models with LiBERO algorithm that we introduced about 10 years ago. When you start with structures and then you generate a lot of conformers, like very small variations, like shimmering sort of the structure, you can generate hundreds of conformers and test each conformer with known ligands and how well they are separated from decoys, building these rock curves, and basically it gives you optimization function for improving the quality and reliability of the model for screening. So you can target several different conformations, and that can be merged into 4D docking. So there are ways. This is also a way to take into account some flexibility on receptor. Okay, so this is great, but what really sort of moved the needle in the last 5 years is expansion and basically like combinatorial explosion of the space that you can use for screening. And so it's, we had like 5 years ago, we had only like about 10 million on-shelf compounds, less than 10 million. And now we can grow it into billions and trillions of compounds, and this is also combined with enormous resources that are accessible on clouds for computation. So we actually run 500 million compounds like straight docking in 24 hours on Google Cloud. but I will show you you don't need this in most cases. So the growth of the libraries. So what actually happened, and thanks to ChemSpace and REAL and Enamine, this was available sort of a few years ago in stock collections that are made by custom synthesis and accumulation of molecules, and also some attempts on generative space where you can sort of dream up molecules from like almost unlimited raw chemical space, but you can dream up molecules during using certain rules. Like for example, this like graph way of like generating all skeletons and then introducing heteroatoms, and with this you get some chemically meaningful molecules, but you never know what is what would be like synthesizability of this molecule in real life, and how much they would cost even if you able to synthesize it. What are the sort of reagents you need to use, and so on. So the on-demand spaces, and specifically real space from ChemSpace and Enamine, they overcome it by using known and well optimized reactions that can be 2 or 3 component, hundreds of thousands of building blocks that can be combined by these reactions to generate on-demand molecules, and this can be into billions, but still having the very predictable success rate, which Enamine guarantees to be at 80% for most of these reactions, and fast synthesis, like in less than 3-4 weeks. So that's really makes a huge difference. And there was like first attempts to use these libraries started like at the level of hundreds of millions of compounds in a library that was like mostly comprised of real compounds by Brian Scheuchert group, where they show that if you scale up this initial virtual library, your virtual hits could be like at the quality that comparable to several to molecules that can be generated after several years of medicinal chemistry, starting from HTS. And if you run here like an additional round of CR by catalog, it like you can move in further. So like let's look a little bit again into our space. So this is like overall drug-like space of 10 to 23 and more compounds, and the millions of compounds in stock, HTS, they're just like a speck in this little speck in this ocean of compounds. And if you find like first hits in this space, there is a long way to lead on drug, and like at each step of optimization, you need to do custom synthesis, basically, because like you need to invent the synthetic route, you need to buy reagents, you need to like do all this trial and error sort of custom synthesis. So the alternative is on real space on-demand virtual libraries, where you have optimized reactions applied to hundreds of thousands of building blocks, and it gives rise to tens of billions compounds. So this one of the most recent libraries was 37 billion compounds. They are virtual but real. I mentioned 3-4 weeks for like lead time for synthesis, 80% success rate. The library is filtered to be drug-like to maintain diversity. So you do not need to worry about this. And this is only like tip of the iceberg, because Enamine has hundreds of millions of tangible building blocks. So you expand like at least more than 1,000-fold building blocks, but because of combinatorics, it expands the library millions and billions fold. So you this gives rise to about 10 to 15 and more tangible real space. So again, if you find compounds in this in real space, for example, they're supposed to be better, like in terms of potencies already and properties if you select for properties. But most importantly, like when you start to optimize it, all the optimization steps are within the same on-demand space, like at least several steps that can lead you, give you way to lead and like almost all the way to drug, and then polish it up with some medicinal chemistry, and you get drug candidate. So that's sort of the vision how the real space can be applied, and many pharma and biotech companies are actually working on this and having first great results, and there are several followers of this as well for the effort that started by ChemSpace and Enamine. Okay, so back to computational. To screen 10 billion compounds, just like the straight docking of 10 billion compounds would cost you about $1 million on like cheapest like baseline prices cloud. So this by itself becomes a huge bottleneck. And if you're thinking of like doing something with trillions of compounds, it like gets completely out of hand. Okay, so there were several acceleration, pretty prominent papers in Nature and other journals proposing acceleration to like allow sort of this practical application of real space. And first, one of the first was this paper on VirtualFlow, where basically going through stages from very crude and fast, like shape-based screening with like less than second per compound to like more elaborate and more accurate screenings, and they did 1.4 billion compounds with this. Another approach exemplified by MolPAL is starting with the whole library, make some iterations between docking and machine learning. So basically, you select a random subset from the whole library of billions of compounds, like say 1 million. You dock them, you train a machine learning, deep learning model. You use this model to select again from the whole space and redock it and do an iteration of this and coming up with hits. It works. It gives enrichment on iterations. And there are several like similar approaches from Jens Carlsen lab, from MolSoft. So sort of with different names and different twists to this, but all of them using this sort of combination of docking and machine learning. It works. The only limitation for this, it scales linearly with number of compounds. And I haven't seen a work like using these approaches that would go beyond like a few billion compounds. So that's real limitation. Now, our idea was to use intrinsic chemical modularity of real libraries to speed up screening combinatorially. And so the idea is that if you have like two attachment points, two component reactions, the full enumeration would be, and you have like 1,000 of R1s, 1,000 of R2s. The full enumeration would be like 1 million compounds. But if you instead enumerate only one side and find the sort of best hits for like part of the pocket, part of the molecules, assuming that sort of the basic concept of fragment-based discovery, that part of the pocket, part of the molecule interaction should be representative. It gives you sort of an anchor interaction for the whole molecule. Then you can enumerate the second half and find the best sort of fit for this, like other parts of the pocket. The speed up of this is enormous because, like for the examples I give here, it will be only like 2,000 of dockings compared to million. And the difference is even more striking if you use like 3-component reactions. That's where the like main growth of real spaces is. Then you have like 1 billion on this side and just 3,000 of this side. So this is like really with scaling of the whole system, the difference between like full enumeration and modular enumeration, iterative enumeration, it grows with the size of the library. Okay, so this was implemented in V-SYNTHES, and I mentioned it was published in January last year. Virtual SYNThon hierarchical enumeration screening, and sort of the idea is like assembling the puzzle in the whole, like in the whole of the pocket, not pre-assembling it and trying. It works by taking first of all taking the chemical space and like with knowledge of chemical reactions and reactants, synthons. Enamine generously shared this information with us. So you can generate what we call ML library, minimal enumeration library. So basically, making the 11 billion compound space, squeezing it into like about 600,000 minimal enumerated compounds, fragments. And so you enumerate one side of the molecule and you cap the other sides because you don't want to leave like reactive groups on the other side that can skew and disturb your docking. So you want these ML fragments to be as close chemically to the final molecule as possible. So that's why we are capping with either methyl group or phenyl group. So with this, you get this minimal enumeration library, you dock it into the pocket, you select the best compounds, the best fragments that best like 1 to 10,000 fragments. You enumerate them on the other side, coming up with about 1 million molecules. Again, these numbers are parameters. You can change them, but that's what we used in this particular paper. And then, so you have this million compounds and you dock these full molecules into the pocket. That's a process for 2-component reaction. For 3-component reaction, you do exactly the same thing, like enumerating the third attachment point, like with one more additional steps. And if you have 4-component, like one more additional step. And in the end, you have you just dock the it's totally usual docking with compounds. Okay, so to have idea sort of how like some internal benchmark, we compared subset from like this 1 million fully enumerated subset after the steps of V-SYNTHES with just randomly selected million molecules from the whole space to see what enrichment we are getting on this stage, and it turned out it is pretty significant. So 40 times. So we have like higher scoring compounds. We have more of them, and the further we go into higher scores, the higher the enrichment. So we use enrichment at 100 compounds, selection of 100 compounds, assuming that you take like 100 compounds into screening, into testing. It also turned out that score is not the only, that we can squeeze some more information from the binding pose. For example, so this is the attachment point, the cap, and attachment point for the second fragment. If it's sort of stuck in the corner of the pocket and there is no place to grow, we call it non-productive pose, and we just remove this fragment from the library, even if it's high scoring. And this gives another several-fold gain in this enrichment factor. So finally got 250 for two-component reaction, 450 for three-component reaction. This was tested on cannabinoid receptor, CB2, getting like synthesizing and testing 60 compounds, 14 of which actually had one better than 1 micromolar potency, and that's something that we never saw with screening of like smaller libraries. So we usually like judge the sort of results of screening and success rate by this threshold, 10 micromolar. But like in this particular case, most of the compounds that were active, they were active at better than 1 micromolar, and some compounds like going into 100 nanomolar range. And we selected the best and done, as I mentioned, an analog by catalog or SAR by catalog, basically in the same ocean of on-demand compound. And we got like for this one series, we got several single-digit nanomolar compounds. So basically like gaining another 100-fold in potency. And this compound is already very selective to CB2 as compared to CB1. Sort of, they're also very drug-like. So the problem with cannabinoid pocket is it's too hydrophobic. So usually you get hydrophobic ligands, and this is not the case. They have very decent log P values. Okay, so we also compared it to like standard VLS subset of the library, and the V-SYNTHES worked 75 times faster and got 6-fold better results like for this screening. In the same paper, there was also test on rock kinase, and we only ordered like 11 compounds, 6 of them were hits, and one of them, I think we got lucky here, was like right off the bat, it was in the nanomolar range. Okay, so how this develops now? So I already mentioned the real space is growing. So most of the studies that like I will show now are done with this description of the library of 31 to 37 billion compounds, but it can grow even further. For selecting of productive pose, we developed a new automatic algorithm called CapSelect that uses geometry of the pocket, and basically it evaluates how much space is left for the compound to grow at the second or third attachment point. So, and if it's blocked, it's non-productive pose. Finally, we are optimizing this V-SYNTHES system with some elements of deep learning. So one of them is we're using deep learning based scoring functions as an orthogonal score to like energy based scores, and this seems to be very helpful because it is less sensitive again to the specific conformation and small variations in the structure, and it gives us additional enrichment in this course. There is also like new tools that we're developing to go beyond just like geometry assessment, but to optimize the selection of poses and sort of predict which synthons will be productive for second and third enumeration. And this use deep learning algorithms and sort of internal benchmarks that we can generate by docking. Finally, we're using improved property predictions, and I will sort of talk a little bit about this later. Some results of applications for new targets, and I mentioned we we stick to quite a bit to GPCRs. So we screened angiotensin 82 receptor for agonists and with quite good results in testing. We have some like other GPCR screening, but also going into some targets like outside enzymes and showing the so enrichment factor out of our internal benchmark, showing that it remains high, and actually higher because we switched to like a little bit more reliable threshold of 10,000 compounds. So we now select like somewhere here, like at 10,000 compounds instead of 100, because the enrichment is so high that like at 100,000 you don't have much statistics, and the estimate becomes reliable. So actually these are all like big improvements compared to what we had published. We keep developing it with working closely with Enamine, Chemspace, MolPAL, and clouds to like fully automate, to expand it, to scale it up, and to sort of automate also the next steps of SR by catalog. The applications, and like some of them are, I will show, some of them I not. So these are like our interactions, our projects with within USC, and we have quite a few academic partners that use like apply V-SYNTHES for their targets. And some examples of primary assays we have. So that's like results that we accumulated over the year, and there are more. I cannot show like the specific targets, but so all of them are quite challenging. So this one is GPCR, but we go after positive and negative allosteric modulators, PAMs and NAMs, and getting quite reasonable hits there that have been validated. We go after agonists in GPCRs. Agonists are usually harder than antagonists, and we do have quite a few compounds that get very high activation. All these initial screenings are done at 10 micromolar, not like 50 or 100. So in most of them I don't show, but there are validation results with full dose response curves. Orphan GPCRs is always considered to be like a harder target. So this one potential Parkinson target, and we get like 4 pretty decent hits out of the screen. And some enzyme inhibitors, so this is Alzheimer target. So in each of these screens, there are validated hits with sub-micromolar potencies or affinities. Yeah, and we are looking forward to taking them sort of to all the way through optimization. So some general lessons that we learn on performance of V-SYNTHES. One thing that one sort of another validation that I never mentioned is checking the poses of final compounds and see if they are similar to the poses of the parent fragments that sort of got rise to the fully enumerated molecules. And we see very good consistency. So the initial binding pose of fragment, if it like high affinity, high scoring fragment, it predicts the binding pose of like the full. Sometimes it's not very easy to like determine. There's some symmetries. So, but in general, this is the rule. So one other sort of aspect, and we tried to do it like to get rid of, for example, of like mail generation and dock directly like SYNThons or dock the like initial building blocks. This doesn't work very well. It turns out like combining scaffolds with SYNThons is very important for several reasons. First of all, I mentioned that makes this minimal enumeration fragments chemically similar to the final product. So there is no sort of artifact interaction that spoil the screening and sort of mislead it. Second, these minimal enumeration fragments, they are slightly bigger than SYNThon themselves, which brings sort of the this MEL library into the docking sweet spot, where of like 2 to 350 daltons, which is known to be like an optimal size of compounds to get selective specific docking versus like non-specific docking. It is also most critical for larger and more complex scaffolds, like in cycloaddiction reaction, because the cycles like triazole, for example, that results from cycloaddiction, it usually makes some meaningful interactions, and you losing them if you do not do this combination of the scaffold, if you do not do reaction. Finally, it's most critical for 3-component reaction. You just first of all, a lot of them are sort of have a bigger scaffolds, and second, you cannot like these smaller fragments do not predict the binding. Okay, so then another thing that we sort of realized is like when we try to use some other docking algorithms, and there's like much faster docking algorithm that employ pre-calculated docking conformers like dock, or like in MolSoft ICM, there is now like an algorithm that pre-calculates the conformers of ligands. they are not very helpful because you have to, the main advantage of this pre-calculation is you do it once and then you can use it for different targets. You don't have this advantage here because you have to calculate the ligands are new, they are calculated on the fly in iterations of the synthesis, so it doesn't help. The receptor flexibility can be incorporated as 4D docking in ICM, but again, you have to be careful. You do not want to incorporate two different conformers because the error at the fragment selection stage, it's sort of magnified and it can mislead if it goes. Well, and speaking of like difference between 3-component and 2-component reactions, the preference for them is defined by the shape of the pocket. So if you have like a long and narrow pocket, it most likely will be best targeted by 2-component compounds, and if your pocket is more bulky, then 3-components reactions are definitely preference. And the latter case is probably most common and most interesting. So 3-components are really important. Okay, so then optional philosophical part. So what are the bottleneck? Not next bottlenecks in sort of computation-driven drug discovery. So one thing is like keep expanding the libraries. One way of doing it is, and this is sort of sidestepping from general purpose spaces like REAL. You can generate on-demand libraries, and so an example of this was presented in Brian Scheuchert work recently when they they developed a library of 75 millions of tetrahydropyridines, and they screened it and they identified hits because pyridines are a preferred scaffold for certain aminergic GPCRs, including 5-HT that was screened in this case. We also tested this approach by using a favorite sort of click reaction by our collaborator Valery Fokin at USC, SUFEX, and making 2 virtual libraries totaling 140 million compounds of sulfonyl-triazole and sulfonyl-isoxazole. So these 100 million compounds were screened again against CB2, the same as in the published paper. It took sort of the bottleneck here was synthesis because they are not on-demand compounds. It took like 3 years and some effort to synthesize 11 compounds, but surprisingly, half of them, more than half of them were hits, and also we have submicromolar hits here with pretty high affinity. So this approach also works. And this can be merged into like this sort of trial libraries, and new reactions are constantly merging into REAL space by the Enamine and Chemspace efforts. So and the libraries grow. So I mentioned most of like our testing that done over the last year were done on 30 billion compound. So now there is Chemspace came up with XREAL library of 173 billion compounds in probably like 3 or 4 months ago. And just to give you the scale, sort of these are compounds used for HTS. These are like enumerated, fully enumerated REAL library, and this is XREAL. And just to emphasize, so screening of the HTS would cost you like about $100,000. Screening of VLS, including purchase of compounds and like everything, can go as low as like tens of thousands of compounds. I guess Yurii just mentioned this. There is next version of X-Real that goes to about 3 trillion compounds. So with new reactions, mostly focusing on 3-component reactions. So that's constantly growing, and it probably like growing with a rate of like Murphy's law, but instead of doubling every 2 years, it gaining like an order of magnitude almost every year. So that's pretty amazing. And I should say this is not like completely unprecedented. There are like other libraries that being developed like especially so, but you can notice like most of them stay with less than 1 billion compounds. Here there are few that grow well beyond actually billions. These are proprietary compounds from big pharma, proprietary spaces from big pharma. But unfortunately, they are not published. There is no like established success rate for them, so it's a little bit of black box. But people dreaming of these bigger spaces. Why? First of all, there are 2 questions here. One, can we handle actually such enormous spaces? For example, like 10 to 15 compounds. And you should remember, because of the modular nature of V-synthesis, it grows with number of synthons, not with number of compounds. So, and if like it's 3-component reactions, and you have like 100,000 of each of our groups, it will make you 10 to 15. That's sort of ideal case, and you will only need to dock like 300,000 mel fragments to dock. In practice, it's much more because like there are many scaffolds. The distribution of compounds on our groups is not even. So you are talking about like several tens of millions of compounds to dock, which is still manageable. I mentioned we did docking of like 500 million compounds directly on cloud. So at the same time, you don't need to do this because there are like this iterative docking, going through docking and deep learning approaches, iterations between them, and like you can squeeze like 10 million compound docking into like less than million, reducing the size of this. And finally, and we are working on this to implement deep learning like on even deeper level, where we can predict from results of mel docking, we can predict which compounds are most likely have to be enumerated and have to be docked. So there are several sort of levels of this, and so what is the bottom line is we can easily handle 10 to 15 right now with the V-synthesis as it is right now, but it can grow even like several orders of magnitude further with if it's combined with some of the deep learning approaches. Second big question is, so we can use them. The big question is, do we need that many compounds? And the answer is yes, the more the better, because this is in like double logarithmic scale, the tail of distribution, and it's supposed to be exponential, so it's like almost straight lines here. So for million compounds, you are expected to have barely like a few hits, like in 10 micromolar range. If you sort of scale it up 100-fold, you scale up, you sort of move this curve, and you are getting much more hits, like hundreds or thousands of hits, and you can get into sort of the affinities range of leads and drugs. And 10 billions gives you potentially even some drug candidates. You just need to take chances to select them. And if you grow even further, sort of this is supposed to get better. One sort of bottleneck here is, and I'm taking this sort of saturating curves from one of the Brian Shohead papers. The curves, so the higher the scores, for example, in docking, the higher probability of this compound being a hit, like in certain range. But then sort of these curves saturate, and the best scoring compound, the compound that predicted to have nanomolar affinities, they actually are not more likely to be like nanomolar hits. And the reason, there are several reasons for this, which are sort of not necessarily unavoidable. So first of all, there are no very good tools for post-processing results. So we are getting like straight out of like V-SYNTHES screening, we're getting like tens of hundreds of thousands of potential hits and leads, but we do not have like good tools to predict which of them are like even more likely, and to sort of extend the linear growth of these curves here. So like still a lot of visual inspection. I see papers where people mention like 30,000 compounds being eyeballed, sort of. One other like sort of explanation for this saturation of curves is some holes in scoring functions. Again, mentioned in this paper by Brian Shohead. We need to fix this, and there are several ways of doing it. Improving the quality of structures themselves by validation, that should be useful. Then reoptimizing the scoring function themselves, because right now the scoring function we use, they are optimized to select binders versus non-binders using this 10 micromolar threshold. So if the optimization of scoring function is done for selecting like 100 nanomolar binders versus non-binders, this could shift sort of the application to this even higher range. And finally, and probably the easiest to implement, is consensus of several orthogonal scoring functions. For example, we are using, I mentioned we're using deep learning based function RTCNN from MolSoft, and that allows us to be a bit fit. Oh, okay, not finally. Finally, the you can take the best like focused library hundreds, a few hundreds or thousand compounds into free energy perturbation method that in many cases gives like better predictions of accuracy. Okay, and then sort of a little bit to elaborate on this. So you still get a lot of candidates from 10 to 15. So you expect to have them. So the if you have a hit rate like 10 minus 5, and that's what we expect for like hits, still 10 billion of potential hits. So if we select sort of in target affinity score, we would get 10 billion candidates, and they are all equally possible to be hits, right? So why cannot you apply like a second criteria and apply like another select again 1 in 10 in 100,000 compounds, and you will still have 100,000 candidates with the second property of your choice to improve. And this property can be ADMET PK properties, or it can be target selectivity, which usually not taken into account at the first stages only in optimization, but right straight off the bat, if you have a way to predict targets off target, for example, binding to closely related receptor, you can select for this right away, or you can select for polypharmacology. And I've seen already like a couple of preliminary studies on applying giga-scale screening to two completely different sort of targets and getting some hits there. Okay, so speaking of drug discovery in general, we do have like one of the examples when this program started like 5 years ago, so it was not exactly V-SYNTHES, but it started with 500 million compound screening, finding initial hits, finding analogs, and optimizing it, getting into like medicinal chemistry, though they were very simple reactions. So our chemistry partners, they synthesized like hundreds of molecules, getting to nanomolar range very quickly, and then spending another almost 1,000 compounds on optimizing ADMET PK properties. And now we have PK candidate, preclinical candidate that is more efficacious and more it has better half-life than the previous clinical candidate and have great bioavailability. And hopefully we can go into, so we're running preclinical studies now, and hopefully we can do IND in about a year. And in general, sort of this can be generalized. The applicability sort of the enormous chemical space, like on-demand chemical space and tangible chemical spaces, to identifying hits, to optimizing them, QSR, and like all the way to final medchem and preclinical candidate. And this can dramatically, supported by proper computational tools, this can dramatically improve the timeline for drug discovery before clinical trials. Okay, so sorry, like a little bit over time. So I would like to thank my lab and especially the power couple of Anastasia and Arman Sedebekov, and our collaborators at USC, and great collaborators all over the world. And most and foremost is we want to thank, and we are very grateful to Yurii, Moroz, and all people in ChemSpace and Enamine who developed this field of on-demand libraries and keep going with like really like breaking speed to improve and expand it. And our lab is looking for postdocs, so if you have somebody in mind, please drop me a line. Okay, thank you for your attention.
Yeah, thank you, Seva. Thank you for this really great presentation. I have just a couple questions. And you know, that's in the sake of time, we just we'll just stick to what we already have from our attendees. Do you visually select hits?
Well, I already mentioned like eyeballing is a secret sort of sauce that all the virtual screening people use. Some more extensively, some less. We do have pretty like extensive and automated sort of post-processing algorithms that bring us from like usual selection of like 100,000 of compounds to probably like 1,000, like half a thousand, and then we do eyeballing them. We do visual screening. And in many cases, when target is challenging, we also like to look into intermediate states, like of how the MEL library is screened. Does it make sense? Because like if something is wrong with the model and the like it gives some strange results on the initial stages, the whole thing could go wrong. So yes, there is still quite a bit of element because structures are not ideal, the structural models are not ideal. That's the main sort of reason for this.
Yeah, thank you. So what other protein classes except GPCRs have you used for virtual screening?
Well, I mentioned sort of within GPCRs, we're expanding it to agonist and to orphan GPCRs, which are and allosteric modulators, which are pretty challenging by itself. Outside of that, I cannot disclose the targets, but these are enzymes, like mostly. So. We do not want to, like we had one example of kinase, like in the paper, rock kinase, and we know it will work, but there is a lot of activity in like generative model and deep learning based models for kinases. So we actually do not, we think that sort of that with the level of knowledge of pharmacology for the kinase family, we do not want to go there. It's just like it won't make much difference.
Thank you. So from the V-SYNTHES paper, how is the 11 billion compounds calculated that mentioned in the title of the paper? If you add up all the compounds from each step, the numbers will be few million compounds.
Right. So 11 billion compounds is the space that is represented by our docking. We don't claim that we like dock all of them. But we screen, we select the best. We believe that we're selecting the best hits out of the whole 11 billion compound space by docking only few million.
Yeah.
And that's the point of like all the acceleration sort of algorithms that are developed.
Yeah, and indeed, this is really a breakthrough. And by the way, as we already mentioned, myself and Seva, we have access to more than almost 3 trillion chemical space through X-REAL, and that's available for ChemSpace customers who want to work on V-SYNTHES with us. So if you have an interest in such an activity, just drop us the line. And I think the last question we have is that you mentioned the challenges of selecting compounds from VLS for experimental testing. I suppose you and the VLS community have a wealth of data about compounds that had good VLS scores, but that were not good binders, inhibitors. Can you use this negative data to improve the post-processing step or scoring functions?
Well, that's a good question. In principle, you would like to tap into this information, and like negative data are critical for good training of deep learning models. The only problem here is that all this information will be very target specific. So the compounds that are predicted to have high scores but not actually show any activity, the reason for this is specific for each target. So it could be like one critical residue that sort of moves in this like slightly off in the structure, or it could be like water molecule that it was not taken into account because the structure is lower resolution and it doesn't see molecule, water molecule, but this water molecule, the like penalty for displacing it like 2 kilocalories per mole, and it spoils like all the prediction that target this spot where this molecule sits, where this water molecule sits. So there are a lot of sort of different reasons, and they are unfortunately target specific and even like model specific. So how useful it to sort of retraining scoring functions? Not sure. But yeah, we're looking into possibilities of like using as much information. But where we see like the best way of like implementing machine learning, like together with Docker into the, is like to do training on the fly, sort of for specific target, like see where like what lesson we can learn from like docking of MEL compounds and like retrain it based on this. But again, this relies on scores.
Yeah, thank you so much for the answer, and thank you so much, Seva, for this really great talk, and thank you everyone for attending our webinar today. I think, yeah, with that, I would like to close webinar. And before the closing, I would like again to mention that please follow us on LinkedIn, Facebook, or Instagram to get information about our upcoming events. And there will be upcoming events related to the series of the webinars that basically describe the use and creation of the chemical spaces. Thank you, everyone, again, and have a good one.
Thank you.
Bye.